Going back to finding the closely related organisms to humans, the amino acid sequences for the production of ACVR1 genes of organisms must be sequenced, and their similarities must be compared using BLAST, or the Basic Local Alignment Search Tool, which is an online software used to compare and locate similar regions between biological sequences, such as the nucleotide sequences of certain genes. You must go to the website www.ncbi.nlm.nih.gov/homologene to reach this page. Next, you must type into the search bar the organism of choice, which for us is Fibrodysplasia ossificans progressiva, and enter. You will then be directed to a page that looks like this. Next, we must look for the section which is protein alignments and show pairwise alignment scores, which is a list of identified organisms with the ACVR1 gene and the percent identity of the proteins and DNA sequences responsible for the production of the ACVR1 gene. Here we will now be comparing the percent identities of the proteins and DNA of the organisms and select which pairs have the highest percent identities when compared to Homo sapiens. Next step will be to use the BLAST software onto these different organisms which have the ACVR1 gene. Now we must use the BLAST software, which will direct us to a page that looks like this, which will lead us to this. From here, we must look for the accession number of the organism, which will lead us to this page. Now what we need here in this page is the FASTA sequence, in which what we will do now is to save this onto a notepad. Now it is important to remember to not exclude this symbol right here, else the program will not work. We will then save the file into a .fas file, a fast file, like so. That process must be repeated throughout every organism. After processing all of the organisms within the list, we will now obtain files that look like this in their fast type files. What we will then do next is to open up a software known as Mega, which would look like this. What we'll then do from here is to go to Align and build in alignment, create a new alignment, and since we're using protein sequences, we'll select protein, which will open up a page that looks like this. Now, in order to select our organisms, we will insert sequences, which is these, open, and we will get this. After we've obtained this page, we must now align them, align by Crystal W, select all, and align them for the default sequence. We will then get this. After aligning all the sequences, we will now export the alignment in a FASTA format and name it. After exporting it into a FASTA file, we will then select phylogenetic analysis. After aligning and exporting, we will then exit this page and then go back to the original Mega page, which is this one. We will then select models and find the best protein model. Yes, currently use the active one, which will lead us to this. And select compute, and wait for this process to be done. This process is done in order to find which would be the best model to use in constructing a phylogenetic tree. Once it has finished loading, we will get a page that looks like this. What we'll mainly be looking at is the BIC, and we will be looking for the lowest value, because it is said that the BIC with the lowest value would indicate that it is the best model. We can see from this list, the best possible model is the JTT plus G plus I, which has the lowest BIC number. After obtaining this information, we will then minimize. We will then return to the mega page in order to create the phylogeny. We will then select the aforementioned best model, as here the JTT plus G plus I, and use the bootstrap method with 1000 replications in order to actually find the best possible model. We compute. Been computed, this will appear. This is the final phylogenetic tree of the bootstrap consensus tree, and this one is the original tree. This shows the evolutionary phylogenetic relationships between every organism which you inputted into the model.